Thanks very much, Rory. So I kind of get the easy job in terms of introducing a session, because unless you've had your head very firmly embedded in the sand over the last couple of years, you'll have noticed that the genetic analysis of the UK Biobank population has really lighted up biomedical research in a way that we may never have expected. My name is McVean. I'm director of something called the Big Data Institute in Oxford, and it's been my pleasure and privilege to work with the UK Biobank to help them design this original SNP array, and then to move on to think about how we can look at whole genome sequencing. But I'm also one of the users of the UK Biobank data, and I can't tell you how much fun I've had in terms of exploring what is an incredibly rich source of information. Now, genetics is, of course, one of many types of information that is measured within the resource, and there are all sorts of other high-throughput molecular analyses, many of which we will hear about later. So it's sort of not different in that sense. But what is different about genetics, and probably why it's attracted so much attention, is that within the space of things that you can measure, genetics really does hold a pretty much unique position, because an association between a genetic variant and some trait that you can measure, as long as you've corrected appropriately for the necessary covariance, that kind of correlation points to a causal relationship. And that sort of digging out the gold of causation actually in correlation is something that really puts the downstream biomedical research on an incredibly strong footing. So the pages of journals, scientific journals, have been littered with large-scale studies of the UK Biobank over the last year or two. But beyond that, actually, the genetic analysis that we have to date is also going slightly over from just the biomedical research into thinking about how we can use the analyses of these really big populations to really test questions about how we can use genetics within clinical research, within clinical practice. So we're going beyond just the discovery program to actually think about how we might use things like genetic risk scores to stratify people into high risk or low risk, or to think about how we might look at subtypes of disease, or how people might respond to particular types of treatment. So what's kind of been the most amazing thing, I think, about the last year or two of research is just how many different avenues have been opened up by the release of the SNP data for the UK Biobank. So that's everything from the study of complex traits to the analysis of polygenic adaptation, from polygenic risk scores through to Mendelian randomization. People are really beginning to sort of open up new ways of thinking about data, and in particular how you can make use of the fact that we have so many things measured on so many individuals. And just one example of that, there was a paper last year on the genetics of neuroimaging. It was the very first tranche of the neuroimaging study. But what that paper showed, I think, was just how much richness there is, just how much additional information we can begin to mine about how genetics shapes human biology and the implications of that for disease. That is sort of pointing the way to what we expect in the future. Now, of course, you can never have enough of a good thing. So where we are today is not the end of the genetic characterization of the UK Biobank. We're going to hear about how going beyond the SNP data, the exome data is going to sort of reveal many new things about how rare variants, highly penetrant variants within genes can shape human health. But also, we want to start thinking about what would we be able to do if we could actually sequence the whole genome of everyone, and not just the UK Biobank. How does then that feed into how we should think about the genetic characterization of more and more populations? So there's an incredibly stellar lineup of speakers for you this morning, and it's my great pleasure to welcome Sir John Bell to kick us off, who's going to tell us about how the UK Biobank is really shaping our thinking about how to do genomics within the UK.